Hello everyone and welcome back to O-Level Oasis. This is your guide for preparing for your O-Level Biology Cambridge examinations. We are currently studying Chapter 17, which is Inheritance. Today, we will begin with Unit 17.2, which focuses on DNA. In this unit, we will go through the key specifications that you need to understand, so let us begin. Now we will begin this unit with Specification 1, which asks us to describe the structure of a DNA molecule. According to this, you need to understand that DNA consists of two strands that are coiled together to form a structure known as a double helix. Each of these strands is made up of a long chain of repeating units called nucleotides. Every nucleotide contains a base, and there are 4 types of bases found in DNA, which are A, T, C, and G. You are not required to learn their full names at this level. The two strands of DNA are held together by bonds between pairs of these bases. These bases always pair in a specific way, are always pairs with T, and C always pairs with G. Now let us define DNA. As we have already studied DNA earlier, we will quickly recall its meaning and importance. So, the key definition is that deoxyribonucleic acid is a molecule that carries the genetic instructions needed for the development, functioning, and reproduction of all living organisms and some viruses. So we already know about the organization of DNA and how it is arranged inside the cell. Inside a cell, the nucleus acts as the control center and contains the genetic material. Within the nucleus, DNA is tightly coiled around proteins to form structures called chromosomes. This coiling helps organize the long DNA strands and ensures they can be properly separated during cell division. A gene is a small section of DNA found on a chromosome, and it carries instructions for making proteins. These genes are the basic units of inheritance. DNA itself is a double helix molecule made up of 2 long strands. It contains 4 types of bases, which are A, T, G, and C, and these bases store the genetic information. Now let us understand the structure of a DNA molecule. DNA, or deoxyribonucleic acid, is the molecule that carries genetic instructions for the growth, development, and functioning of living organisms. It is found in the nucleus of cells and is organized into structures called chromosomes. A DNA molecule is made of 2 strands that coil around each other to form a double helix, often described as a twisted ladder. Each strand is made up of a long chain of repeating units called nucleotides. A nucleotide is the basic unit of DNA and has 3 parts, a sugar, a phosphate group, and a base. Now here, it shows the structure of a nucleotide and how many nucleotides join together to form a chain. A single nucleotide has 3 parts, a phosphate group, a sugar called deoxyribose, and a nitrogen base such as A, T, C, or G. When nucleotides join together, they form a long chain called a polynucleotide. They are linked by strong bonds between the sugar of one nucleotide and the phosphate of the next, forming a sugar-phosphate backbone. The bases extend from this backbone, and their sequence carries the genetic information. Now let us understand how the DNA structure is formed. Each nucleotide contains a base, and there are 4 different bases in DNA. These are A, T, C, and G. Nucleotides join together through their phosphate groups to form long chains, which can be thousands of units long. Along each strand, the sugar and phosphate form a constant backbone, while the bases are the parts that vary. The DNA molecule consists of 2 such strands. These strands are held together by bonds between pairs of bases. The bases always pair in a specific way, are always pairs with T, and C always pairs with G. This fixed pattern is known as complementary base pairing. DNA is built step by step like a structure formed brick by brick. It is the molecule that carries genetic instructions in all living organisms. The basic unit of DNA is called a nucleotide. Each nucleotide has 3 parts. It contains a sugar called deoxyribose, a phosphate group, and a nitrogen base. The sugar and phosphate form the structural framework, while the base carries the genetic information. There are 4 types of bases in DNA. Adenine pairs with thymine, and guanine pairs with cytosine. This specific pairing follows a fixed rule. When many nucleotides join together, they form 2 long strands. These strands twist around each other to form the double helix shape. The sides of this structure are made of the sugar-phosphate backbone, while the middle part consists of base pairs held together by bonds. This arrangement is known as complementary base pairing, where A always pairs with T and G always pairs with C. This DNA is often described as a ladder that is twisted into a spiral shape. First, think of the ladder structure. The sides of the ladder, also called the backbone, are made of repeating sugar and phosphate units. The steps of the ladder are formed by pairs of bases joined together by bonds. In these pairs, adenine always joins with thymine, and guanine always joins with cytosine. Now, this ladder does not stay flat. It twists around itself to form a spiral shape called a double helix. This twisting helps a very long DNA molecule to fit inside the small nucleus of a cell. So, this is how the double helix is organized into specific parts. The outer part of the DNA molecule forms the sugar-phosphate backbone. This is made of alternating sugar called deoxyribose and phosphate groups. It acts like the supporting rails of a ladder and gives the DNA its strength and stability. Inside the structure, the nitrogen bases form the rungs of the ladder. There are 4 types of bases: adenine, thymine, guanine, and cytosine. These bases follow a specific pairing rule called complementary base pairing. Adenine always pairs with thymine using 2 bonds, while guanine always pairs with cytosine using 3 bonds. This fixed pairing helps hold the 2 strands together. The 2 DNA strands run in opposite directions and twist around each other. This twisting forms the characteristic double helix shape, which is the stable spiral structure of DNA. Now let us bring together everything we have learned about the structure of a DNA molecule in a clear overview. DNA can be understood as a ladder-like structure. The sides of this ladder are formed by a sugar-phosphate backbone, which provides strength and support. The rungs of the ladder are made of pairs of nitrogen bases that join the two strands together. This entire structure is twisted into a spiral shape called a double helix. The two strands are held together by weak hydrogen bonds between the base pairs. The order of these bases forms a genetic code, which contains instructions for building proteins. These proteins control the structure and activities of cells, so the DNA code ultimately determines how an organism develops and functions. A gene is a specific section of DNA that carries the instructions for making one protein. Differences in base sequences lead to different proteins, and this is what causes variation among organisms. Again, this diagram shows a section of a DNA molecule, which is arranged in the form of a double helix made up of two strands. Each strand has a sugar-phosphate backbone, and nitrogen bases are attached to it. These bases connect with the bases on the opposite strand to form base pairs. The four nitrogen bases carry genetic information and always pair in a specific way. Adenine always pairs with thymine, thymine always pairs with adenine, cytosine always pairs with guanine, and guanine always pairs with cytosine. For example, a sequence such as adenine, cytosine, guanine is part of the genetic code that stores information in DNA. The main points to remember are that base pairing is specific, with adenine pairing with thymine and cytosine pairing with guanine. The sugar-phosphate backbone forms the outer structure, while the bases are found in the center. DNA acts as a chemical blueprint for living organisms, and understanding base pairing is important for understanding how genetic information is stored and passed on.